Hello, I am Amrita, Assistant Professor, Krishna Jayanti University. I am going to discuss a topic of genetic engineering, which is biological method of transformation. So transformation, as we know, is the insertion of an external DNA into a host cell. And there are various mechanisms of transformation. They are physical, chemical, as well as biological. Today, in this discussion, we will focus on the biological methods of transformation, and we will be taking inspiration from the natural way of biological transformation, which is seen in crown gallbladder disease in certain plants. And the plant that I have shown you here is infected by a bacterium referred to as Agrobacterium tumefaciens. And this bacteria has a TI plasmid with a tDNA region as well as the virulence genes that helps the entire plasmid along with the tDNA region to enter into the plant and infect the plant. And the result is this tumor-like region that is developed in the plant. In this slide, I want to show how this entire process occurs. So the TI plasmid is the circular plasmid that we see here. This is has a region called tDNA region as well as the restriction cleavage site. And it also has virulence genes that helps it in the entry of this plasmid into the plant. This particular region is exploited by the synthetic mechanisms in the lab to insert the DNA of interest and then allow it to enter into the plants. And once it enters into the plant, it infects the plant. The new genetic material is taken up by the plants, and that is how the genetic transformation of the plant occurs. So this is one mechanism using Agrobacterium tumefaciens. Uh, and this is one of the commonly used method in the labs. The second method I want to discuss is that of viral mediated transformation. Now, viruses also, as we know, have the tendency to insert its DNA into the host cells. And there are various mechanisms that is already known, lytic cycle, where it breaks into the host cell and that it lyses the host cells and releases the genetic material. And in lysogenic cycle, the viral DNA is inserted into the host DNA and becomes a part of the host DNA and then multiplies. So this has also been employed to insert the DNA of interest, and many a times the viruses are made to take up the gene of our interest, and once it is taken it up, virus feels that it is its own genetic material. And the same process occurs that it once it enters into the host cell, it mm, provides its own the DNA that was there in the virus's capsid is taken up by the host cells readily and becomes a part of the host genome. So there have been many ways in which this mechanism has been used to cause cancer-like changes in cells by introducing oncoproteins so that we better understand how oncogenic uh, regions they code for different proteins, oncoproteins, and how they cause the tumor-like conditions in humans. And then once we have that idea, we can use it to test our drugs, and we can also use it for gene therapy and research. And viruses are also being engineered into vectors to deliver beneficial genes into the host cells. So this entire process is called as viral transduction. There are various viruses that are known to carry out this function: adenovirus, lentivirus, herpes simplex virus, and each of these viruses have their own properties. For example, adeno-associated virus is a single-stranded DNA, whereas herpes simplex virus is a double-stranded DNA, and lentivirus is an RNA. Their capacity to carry the genetic material is also very different, with herpes simplex virus having the largest, extraordinarily high capability to carry large size of genetic material. And the speed of expression is also different, and duration of expression also becomes different in all the cases, and it is highlighted in the slides. Now, once the transformation has occurred, we also need to know that which are the cells where the transformation has occurred and whether the transformation has occurred at the right place or not. One of the very common methods that we use for this purpose is blue-white screening. Now, blue-white screening is a very common screening which utilizes LacZ operon in E. coli. Now, LacZ operon has several genes that code for proteins that is activated by lactose, presence of lactose, and that utilizes lactose for the metabolism. So they produce an enzyme called as beta galactosidase. And if this particular transformation is occurring at the right position, then often the operons get disrupted. And once the operons get disrupted, the beta galactosidase enzyme is not produced, and the lactose or analog of lactose is not digested. So what we do for this method is that we use an XGAL. XGAL is nothing but chromogenic material that can be digested by beta galactosidase enzyme, and it is hydrolyzed to form 5-bromo-4-chloroindoxyl, which spontaneously dimerizes to produce an insoluble blue pigment. So if beta galactosidase is produced, then XGAL is hydrolyzed and a blue pigment is produced. Now, let's say that we carry out transformation in the XGAL region, and there is an insertion of an external genetic material in the same region. Then what do you think would be the outcome? The beta galactosidase gene that code for this protein will be disrupted because of transformation. Hence, the enzyme will not be produced. And if the enzyme is not produced, XGAL is not digested. As a result, the blue pigment is not produced and the pigment remains white. So we get an idea if the transformation has occurred, the bacterial culture that grows is white in color. And if the transformation has not occurred, then the XGAL, the beta galactosidase will be produced as usual, and XGAL will be digested into blue color. So here I have shown in a Petri plate where the reaction has occurred. So we can see different cultures that have grown in the Petri plate. So wherever the blue color exists, that means that the beta galactosidase coding genetic region has not got disrupted by transformation, and the transformation has not occurred. And wherever the white colored culture exists, that means that that particular genetic region was disrupted by transformation, and because it was disrupted by transformation, the XGAL was not converted into blue chromogenic color. Now, one more thing I want to highlight is that what if, let's say that the gene of interest exists. That means that, but it is not inserted into the right region. Let's say we have the DNA, okay, and we also have we have the DNA of interest that we wanted to insert, but we also have the plasmid where the transformation has not occurred. So to ensure that we are only selecting those regions where the transformation has successfully occurred, we have a selection marker gene also. Here we have an ampicillin resistant gene. Now, only when there is, we want to only select those where this plasmid is there. So just the gene of interest will not survive in this area, because this Petri plate will have ampicillin, and only when the plasmid, we only want those bacteria with the plasmid and with the transformation. So for the bacteria to survive in the ampicillin existing plate, there is a need for the bacteria to have this plasmid, ampicillin resistant plasmid. So we use to ensure that there is no false negative, we ensure that we have the ampicillin resistance gene as well as we have the LacZ gene, and we have the cloning region within this gene. Here is the diagram that shows that what are the processes that we may observe. So for example, here is an insert within LacZ gene with ampicillin resistance as well. In that case, we will have white colony. If the insert is outside LacZ region, then also we will have blue colony. And if the transformation has not occurred, then also we will have blue colony. Lastly, there is also a mechanism using PCR. If we want to know immediately and we do not want to wait, we can also have a colony PCR. We can take this colony and we can carry out the PCR just to check if that particular transforming gene, the gene that was transformed, exists or not. So that also can be undertaken if we are short of time and we need to know immediately whether the gene of interest has been inserted into the bacteria or not. So the method that is followed is we pick a colony, we lyse it, and we add it directly in the PCR mixture, and the high heat of the PCR will itself lyse the bacterial cell, release the plasmid DNA to be used as template. So no need to lyse before. Even in the PCR, the lysing can be done. In other mechanisms, we lyse the bacterial, extract the DNA, and then carry out the PCR. In colony PCR, the heat of the PCR method takes care of this issue. We amplify it, and ultimately we analyze the size of the PCR product in the gel electrophoresis. That gives a very good idea about whether it exists or not. And there is a variation of PCR results outcome in the transformed cells, in the non-transformed cells. So here we know how there are different methods, biological methods also of transformation, and whether the transformation has been successful or not can be observed either using blue-white screening, colony PCR, as well as by ampicillin resistance genes, etc. So this is all for today. Thank you very much.